Welcome to BioClass. Today we'll discuss the different properties of enzyme and the classification of enzyme. The different properties of enzymes, it is already it is told and given in the previous classes. Today let's see by taking example. Take an example here, one chemical reaction, here the carbon dioxide and the water, and here the carbonic acid. Here this reaction we can use without enzyme also and using this carbonic anhydrase enzyme. Okay. So, what will be the rate of in each cases? If the if without enzyme this rate is 200 per hours, 200 molecule per hours that it can be formed. But if it is if we can use that carbonic anhydrase, then it will be 10 million times, 10 million times, and the rate will be here 2.16 into 10 to the power 9 molecule per hour. Just imagine that it increases the rate, increase the efficiency of the reaction. Like catalyst, this is also, and huge turnover rate is there. Huge turnover rate also it is there, that you have to understand. Next, concept of activation energy. Concept of activation energy. What is activation energy? Activation energy is the energy which needed to start a reaction. Okay. This is when an enzyme is used as catalyst, which means it increases the rate of reaction. Now let's see this graph, then we can understand better. Okay. Look at here. This is the reactant, the same carbon dioxide and water vapor, and this is the product. Okay. So energy level one initially it is high and the product it is less. Now without enzyme, if it is given, then activation energy, activation for the starting, that energy is much higher when it is energy is not there, and when energy is there, the blue line, you can see that blue line, that is the activation energy with enzyme. When it means that if it is the amount of the enzyme, this is the amount of the energy, then it will be much lesser. Then this is the energy which is the without enzyme. Ultimately, that products that is we got. Previously we have seen that number of the molecule, the rate, and here also the in case of the energy activation, also the enzyme is much efficient. Next part is nature of enzyme action. Nature of enzyme action, that how enzyme is acting on substrate. Okay. Here is one lock and key model. This model it is proposed by German chemist Emil Fischer in 1890. So there is huge similarity. So from that we can very easily understand that enzyme, the nature of action, enzyme action. Let's see. Here is a substrate. Here is a lock. Here is the key is the enzyme. Here is the active sites of enzyme. Active sites of enzyme is very specific. Enzyme also very specific to the substance. So here lock and key also. For every lock, the key is specific. So it, it indicates that here this is also the huge similarity. This is the active sites. You know that in lock and key, this active sites means this sites, if anyhow it is destroyed, then this key will work. In case of enzyme also the same. Active site, if it is destroyed, then also it doesn't work. Now when after that, if we want to open it, this lock, so we have to insert. So it means that enzyme substrate complex. When it is inserted, now the reaction, it will start, and it will starting the reaction. Now next, when it's open, see that this two part, suppose this is the product, this is also product, I mean one is product and this is byproduct. So product is there and enzyme remain unchanged. Okay. So we are getting the close similarity in enzyme and the lock and key enzyme substrate. One thing, another similarity, again we can lock it with the same key. So it reverse, it goes this side. In case of enzyme also the same. Enzyme also you can the substrate enzyme substrates complex and the products. So 4 steps are there. The first here, the substrate bind to the enzyme, then enzyme substrate complex will form, then the reaction it is done, so the product will form, and fourth is the enzyme remain unchanged, and again this it can react it. So this is the enzyme action that you got. Next part is factors affecting enzyme activity. The different factors like pH, like substrate concentration, like temperature, like enzyme concentration, how it can affect that enzyme activity. So here we can see that the pH, if it is increased, it means that from the neutral to the base, it reaction rate, it is in certain optimum. It is depend upon the enzyme. Enzyme pepsin can react in pH 2, acidic medium, and the several other digestive enzyme in the basic medium. So one certain optimum level is there. It means that this level, if will not reach, then the rate of the reaction it will less. If it is more, suppose in 8 it is here, and in 11 also it is here, it should be 10. Okay. In case of substrate concentration, if we concentrate that substrate, so concentration will increase, that the rate also increase, but one certain level, if we increase the concentration, then also the rate will not increase. So one certain time that is increased, after that it is straight line, it means that the here rate is not increasing, but the concentration we are increasing. One saturation point is there. Temperature, if we see this, the temperature, one certain temperature, normally it is our body temperature 37 to 40. So one optimum temperature it is there. Okay. So in low temperature that rate is low, in the high temperature also it is reduced. Okay. And in one optimum temperature, suppose here it is 37 degrees Celsius, it is maximum, and and around 37, so 35 to 40. So it has this rate as the maximum. And low temperature also less, and high temperature also less. Enzyme concentration, if we increase, then what kind of increase we can see that in rate. Here we can get the straight line. It means that if we increase the enzyme concentration, the rate will increase according to that. Okay. So from this graph we can able to understand. You have to, this is a very important part, you have to keep in mind all this. Next part is inhibitors of enzymes. There is some substance which resembles that substrate of enzyme, and this substrate it is bind by the active sites of the enzyme. So the original substance remain unused. Let's see that here it is succinic dehydrogenase 1 enzyme. This is the real substrate. You can see this is the real substrate. Here one inhibitor, which is the active sites are resemble same. Malonic acid, it is acting substrate and it fits over there. So that real substrate, it is unused. This is called the competitive inhibitor, competitive type of inhibition. All right. Next part is classification and nomenclature of enzymes. Enzymes are classified and grouped into few groups. You can see that oxidoreductase, transferase, hydrolysis, lyase, isomerase, ligase. Here is in your book also in the brief form that it is given. So here I am discussing that the basic things, whatever if here it is, suppose oxidoreductase, here transfer of electron, it means the removing or adding the electrons from the structure. Suppose alcohol dehydrogenase, from the alcohol to aldehyde, when it converts, that electron it is can be transferred. In transferase that different functional group like carbon group, nitrogen, sulfur, phosphates containing group can be transferred. It means that if from AB is there, then here the C and BC, that this kind of transfer it can be there. Hexokinase, the cytochrome oxidase, glutamate dehydrogenase. So different example. Hydrolysis is simple, that in the enzyme with water it is cleaved. Several that cases in enzymes in our body, that is hydrolysis enzyme. It means that here AB, the water is there and the enzyme is there, and it breaks this bond. The trypsin and other enzymes. Lyase, here it is, here also the cleaving the bonds, but here water it is not there. And here it is, look at here, it cleaved and it formed in some other. Suppose decarboxylase, the different kind of pyruvate decarboxylase. Isomerase it forms the isomers. Suppose glucose to fructose. It means that just see that A, B, C, D, and it converts into DC. Only some structural change, but basic both are there. I think you know the isomer. So here the malate isomerase. Now ligase, also called the synthetase. Here that the hydrolysis takes a high energy phosphate from the new bond. So here it will join here, not cleaved. AB, one bond here will form. Next, and the last part, that is a cofactor. So enzymes are composed of several polypeptide chain, that is basically protein, but there is some non-protein part, non-protein part also, that's cofactor, and these are mainly that coenzyme or the prosthetic group. Coenzyme and the prosthetic group, about this, the prosthetic groups, they are they are organic compound that distinguish from the other cofactor, and they are tightly bound with the apoenzyme. Apoenzyme is the protein part of it. I think this structure is already given in the first lesson. Okay, and it is difficult to separate, like calcium ion, magnesium ion, sodium ion. Cofactor are also organic compound, but their association with the apoenzyme is not that, it is loosely bound with that, and it can be easily separated, like NAD plus, ubiquinone, and cytochrome, all these things. Okay. Okay, so that is about that enzyme that you have to prepare nicely. Thank you.